Welcome back from coffee. This is me at Sydney Airport a few weeks ago. I'm, as stated, professor in tree ecophysiology. I'm also working for a company called Arevo, a small startup company that I will tell you more about at the very end of the presentation. The title of my talk, Feeding Forests with Amino Acids, requires some explanations. And this is like a very short version of a long career in research on plant nutrition and what it has led to. But I think a good starting point is 110 years ago. So 110 years ago, this person, Fritz Haber to the left, he invented a procedure by which you can capture not carbon but nitrogen from the air. By combining it with hydrogen gas, you get ammonia. And I would say few, if any, inventions have had such a dramatic impact on humanity. And there are many ways of describing this, but this is one of them. So here we see the growth of the human population, the blue curve here. And, we see also the rise of the production of synthetic fertilizer, what we call the Haber-Bosch process. And the red line actually depicts what the population maximum would have been at had it not been for Fritz Haber and Carl Bosch. So this was really the starting point. For me, we have this immense industry. We have 120 million tons of synthetic fertilizers produced every year, actually contributing quite significantly also to carbon emissions. And as with all great inventions, there is a downside, and this is one of the downsides. Here we see the nitrogen use or the fertilizer use efficiency in agriculture and in forestry. So the global average in agriculture is just below 50%, meaning that 50% of the fertilizer that are added ends up where they are supposed to end up. If we look at forests, then we know that the best way of getting forests to grow more is to fertilize them. We can add 100, 200 kilos of nitrogen per hectare and get a really strong growth response. But we see that only a minor fraction of the fertilizer that we're adding actually ends up where it's supposed to end up, in the trees. And this was really the point of departure for my research group when we started to look into this quite a few years ago. And the first question, well, a question that you would think should have been answered long time ago, but we actually looked into this really deeply. What are the sources of nitrogen available for forest trees? And to get an answer on that, we had to go all the way back to really developing new technology. And some people think this is disgusting, but really this was an eye-opener for me a few years ago when I attended a conference that a type of conference that I usually don't attend, talking about neurotransmitters. And I realized that this technology actually, with the small membranes that they use to get the information on neurotransmitters, in this case, a rat brain, they share many features of plant roots, both the size and some of the functions that plant roots have. So a lot of work in the lab, and then instead of bringing soil samples into the lab, we brought the lab out in the forest, and it looks like this. So we've done a lot of research trying to understand what are the sources, what's the basic foundation of nitrogen in forest soils, especially in boreal forest soils. So if we look at standard technology where we take samples into the lab, this is what we get. We have a very strong dominance of ammonium. We find some nitrate, and we find a few of the organic nitrogen forms, especially amino acids. But from this, you would assume that forest trees, they live on ammonium. But when we bring the lab out in the forest with this neurotransmitter technology, we get really the opposite result. And we can show that most of this is actually produced from that. It's an artifact during handling of soil. So basically, what we now know, or what we think we know, there is always room for improvements and new data. But if this is basically how the soil nitrogen availability looks like in forests, well, the next question, what is available? And for me, it's a little bit strange to say, well, it took me 20 years to put this green arrow here. Oh, sorry. But that's the truth. To really show that in this large group of substances, plants actually acquire a lot of nitrogen. And we've done all kinds of research to actually prove this. We've done field research, we've been into the molecular underpinnings of this process, so we have a good understanding of the process. But then we started thinking about applications, and for specific reasons I started thinking about fertilizers then. We have nitrate and we have ammonium. Ammonium nitrate is standard fertilizer, together with urea, dominating the nitrogen fertilizer industry totally. But still, a sort of limited number of compounds. Here we have 20 protein amino acids, and we have something like 200, 250 other amino acids out in the soil. So we have a large group of substances, and if we want to try some of those as fertilizers, where do we begin? How do we get information about that? Well, the very simple question was, ask a pine seedling. And this is like the choice test, like a smorgasbord for a pine seedling. And we've done this with many different combinations. So here is only 4 different compounds. So it's roots, or actually these are seedlings grown in pots and then supplied with mixtures. And then we look into what do these seedlings prefer. And as you see, they don't like nitrate at all. They take some of this amino acid glycine. They are really good at absorbing ammonium, but basic amino acids, and there are 3 of them, and especially this one, arginine, seem to be the preferred choice. And there are specific reasons for that, but from this, the idea was simply, all right, can we develop a fertilizer based on arginine? And we've been working on that from many different angles, but there's this biological sort of understanding, but there is also this thing that, okay, if this is a fertilizer, we have access to completely different and new type of chemistry. We can produce different types of fertilizers. I'll soon come back to that. Yes, so this is one of the more recent inventions that we have, where we take this chemistry to produce a slow-release fertilizer, a fertilizer that has a long-lasting effect. And in this case, we combine nitrogen in the form of arginine with phosphate. So we get these beautiful crystals. And as I said, these have long-lasting nutritional effects on plants. And so there is a new type of chemistry. There is also a new type of biology, because ammonium nitrate or urea fertilizers, when you add them, you see the responses really directly. You see greener plants, you see bigger shoots, longer needles, or bigger leaves and all that. But with this fertilizer, we see something different. So here's an example where seedlings, again, Scotch pine seedlings have been, we've added either arginine phosphate or ammonium nitrate-based fertilizers. If you would just look at the upper part, this one actually looks better. When we look below ground, there is a big, big difference. And it's true both for the total root biomass, and it's true for, as we see in this graph, the fraction of root tips that actually carry mycorrhiza. So it's not only that we have access to a new type of chemistry, we also get different kind of effect. We stimulate root growth rather than shoot growth. So in Finland and Norway and Sweden, somewhere around 600 to 700 million seedlings are produced every year. I was searching for information on a European level, but I didn't find it. My guess would be billion or just about a billion. So this is a big industry. As Anneli said, I think we try to plant at least 2 seedlings per cut tree. And it's really important that we get them to grow well when we plant them. If we look at the forest rotation cycle, and this is the reason we have actually a circular economy connected to this industry, we see that there is after cutting, we have this period where seedlings have been planted, but they're really small. During this phase, we actually have a net emission of carbon dioxide, so we want to keep it as short as possible. For several reasons. Carbon emissions is one. It's dangerous to be small out in the forest is another, because many herbivores are interested in eating these seedlings. Obviously, this is not true for northern Sweden, where I come from. It should stay 100 years or 120 years here, but you see the principle. So we want to keep this phase as short as possible. Obviously, we want to fertilize them then. We want to add something so they grow really quickly and grow out of this dangerous and sort of negative phase. Problem is that with conventional fertilizers then, slow release type fertilizers, as I said, we stimulate shoot growth rather than root growth. And in a situation where you have dry soils and you have this seedling coming from a really nice environment in the nursery and going down in the soil, then we want the opposite. We want to have strong root growth rather than strong shoot growth. So this negative impact, this is from a study actually in the US, negative impact on lateral roots, that's the roots growing out from the original sort of root structure of the pot. It leads to well, increased losses, basically because they are getting more drought sensitive. When we add instead this fertilizer based on the new chemistry, the arginine phosphate, the slow release nitrogen phosphorus combination, we really see the opposite, a very, very strong stimulation of root growth. And one of the small details here is that we add 0.04 grams per seedling. If we plant 2,000 seedlings, that amounts to 80 grams. That's hardly a fertilization. This is something much more precise, something with hopefully a maximum impact on a very small scale. So here is just a photo showing after one growing season without the fertilizer, which is the standard practice in forestry, and with the fertilizer. And you see some effect on root growth also. Obviously, this one is greener, but the main impact is really on the root and the new roots formed. And this is, as I said, the critical thing for this seedling to survive. So we started this as a research project in 2015. We applied for patents in 2016. And we've started with small-scale tests going from a few hundred to 10,000. 2018, we had tests of 500,000 seedlings out in the forest in combination or in collaboration with most, if not all, big forest companies in Sweden and some in Finland. This year, the commercial phase has started, and we've basically sold out everything that we've managed to produce. Something like 6 million seedlings have been treated with this new type of slow-release fertilizer. And if we go back and look at the first seedlings that were treated, or actually these were treated in 2017. This is how they looked after one growing season, after 2 growing seasons, and we did dig up some seedlings just to look at them after 3 growing seasons. And if we dried them, or actually this was done at the Swedish Forest Institute, we see still a strong increase in root biomass, but then we see also this shoot growth taking off. So we added 40 milligrams of nitrogen here. The difference between these two seedlings is around 40 milligrams. Here it's around 120, and here it's around 600 milligrams. So basically the idea here is to get a kind of priming of the system, get the seedlings to grow the roots out, get the nutrients, especially the nitrogen, that is already available in the soil. There is a lot of nutrients in the soil. Problem is to get the roots out there and capture it. And we had to develop, of course, a deployment system. You don't want to sort of spread these tiny amounts of fertilizers by hand. So in collaboration with the company, we have developed a type of dispenser, which we put on a normal putki planting tool. And this is a 250 ml bottle, and it's good, as you see, for 450 seedlings. So for a planter, this means an extra weight of maximum 0.5 kilo, and it means you need to carry 4 bottles a day with you. It is, well, it's an extra burden, it's an extra thing to add, but still it is feasible for a planter to do this. I mean, you can argue, or you can discuss if this is a good comparison or not. But what I'm saying here is that this activity that we have now of adding 150 kilos of nitrogen per hectare, usually once or maybe twice, we do get like 15, on average, 15 cubic meters extra. And on average in Sweden, that would maybe 3 years of growth. And we think that we can shorten the growth phase, the first growth phase, by approximately 3 years, which would be actually the same. Difference is we spend 80 grams of nitrogen or we spend 150 kilograms of nitrogen. It's not saying that you can't do this if you have done that. It's just saying that if you start early, you do this investment, you actually get a lot of return for it. Yes, and just to end, we started, as I said, in 2015 and 2016, we had the first field studies. And because this product is not available on the market, we had to set up our own production system. And in doing so, we also filed a couple of patents for a specific method to produce these slow-release fertilizers. The last 2 years, we've been working hard to scale up this, and now we're in the phase of actually setting up a full-scale production system that would produce something like 200 tons of fertilizer per year. 200 tons might sound a lot, but actually in the fertilizer industry, it's not on the map. But because the small amount that we add has such a strong effect, we still think it's worth it. So in 200 tons, it's something like 500 million seedlings per year that we can treat. And by that, I think I'll stop there.
Thank you for the extremely interesting presentation, Torgny. You are ready for some questions?
I am, yes.
Perfect. Is the audience ready for some questions? There's one.
One from Umeå, that's
yes. Hello,
nervous.
Torgny. Okay, so you add the arginine at the time you are deploying the seedling, and then it has a very big impact. And I was thinking, actually, it reduces 3 years, the rotation time, if I understood correctly. So have you thought about, or is it beneficial, or would you recommend to add arginine also later on during the growth of the trees?
So I had expected or anticipated another question. What is the cost of the raw material? And the thing is, obviously, that this is a small-scale industry. We use food quality arginine. We have a we have a process. So obviously, this is a much more expensive product than the bulk ammonium nitrate or urea. So therefore, we are targeting situations where a little nitrogen makes a big difference. Hopefully, we've seen, and I've been doing this for a few years, so the price of the raw materials, they are coming down quite rapidly now. And yes, we are doing the tests, but we are not recommending it yet. We have done quite a lot of tests already, but it seems like this first phase, actually I had a couple of extra slides with me. This one, for example, this just shows the nitrogen availability after clear cutting, and these are 2, 4, 6, 7 sites. And what you see here is that after a clear cut, there is a lot of nitrogen available in the soil. So the trick, again, is for us to get the seedlings, or the trick for the seedlings is to get the roots out to capture this pulse. So I'm not sure that an extra pulse would mean a lot. That was a long answer for a short question.
Yes, hi, my name is Niklas von Weimann. I come from Metsä Group. Thank you for a very interesting presentation. My background is in biotechnology, and I know that the microbes love amino acids as a nitrogen source.
Yeah.
So I'm sure you have studied the role of the microbes on the roots, and are they the ones transporting the nitrogen into the roots? Can you elaborate on this?
So, short answer is probably most of it is transported via the mycorrhizal fungi in this case. But we have done quite a lot of research on Arabidopsis, for example, that is non-mycorrhizal, shows the same thing. So it's not that plants are dependent on microbes to convert this. So really they have the capacity. We can see that they express the transporters that enable that for them to take up. But as you're indicating, like an amino acid is the perfect substrate for so many organisms. So in this case, part of the effect that we see is for sure because of enhancement of mycorrhiza. If you add ammonium nitrate, you push back mycorrhizal infections.
Any more questions? Oh, there's one.
Thank you for excellent presentation. My name is Piotr Borkowski. I represent the European State Forest Association in Brussels. My question to you is, I mean, it's a brilliant technology which allows to speed up a bit the production cycle and, for example, the survival of the planted young trees on the regeneration plot in the forestry. But at the same time, have you considered what's the impact on the soil? And then maybe one additional question, how do you see what you are presenting here as a certain solution for the future vis-a-vis the growing pressure from environmental side on forestry, because we are actually very often accused that we are speeding up production cycle, we are not sustainable anymore, we are in conflict of biodiversity. So how to keep the balance, because we can be easily, maybe accused is the strong word, but we can be easily told that we are going towards segregation of the certain functions, which will lead us towards, you know, keeping areas for intensive production, whereas to set aside more and more, and this is actually the current tendency. These days in Brussels, many people are discussing to set aside more and more. Thank you.
Yes, where should I start? I think firstly, we are looking into the microbiome on roots, and we are comparing this product with standard fertilizers. We see big difference in composition, and I would, from that data, it's a little bit premature, but it looks really interesting. So I would say this is mimicking more of a natural situation than, I mean, it's going back all the way where I started. What are the sources of nitrogen that you see in a non-fertilized forest? They are these ones. Arginine is one of the prominent amino acids that you see in soils. So I would still claim that this is, I I don't want to use the word natural because ammonium nitrate, they're also natural, but the negative impact will be smaller. Secondly, I gave a presentation to a small group some time ago, and then one guy raised his hand saying, this looks fine, never call it a fertilizer. Because that will be a negative reaction to fertilizing anything in the forest. And again, if you think of 80 grams, I don't know if you have any ideas what 80 grams per hectare is, you will not find it. You can't measure the impact of this addition of this fertilizer. And again, the whole idea was to get something, a very small amount, to have this big effect. So maybe I just should rename it as, I don't know, a plant nutrient or organic nutrient. That would solve the problem.
Thank you very much, Torgny. Thanks for the presentation. Let's give him a big hand.